So we had this result from optogenetics in mice, and I'm like, wow, this is really cool, and it would be great if we could implement this somehow in humans. And we know from our stuff, like if you globally affect all of the neural activity together, you're not going to get these persistent effects. So can we somehow deliver electrical stimulation in some pattern or in combination with pharmacology to give us the same pattern of cell activity that we got with optogenetics?
Welcome to Stimulating Brains.
Hello everyone and welcome back to Stimulating Brains episode number 24. A few weeks ago I had the great honor to co-organize the OptoDBS conference in Geneva together with Christian Lüscher, who's the main organizer since many years, and we also, if you're more interested in that, talked in episode number 20 about the general concept. The idea behind the conference might best be articulated by a paper that Christian Lüscher wrote together with Megan Creed in Science, and is briefly summarized in a way that you would use optogenetics to understand the key circuits that you have to exactly modulate, for example, in order to treat a symptom. And then once you have understood that, find a smart way of doing the same and exact same by electricity, so in other words, by deep brain stimulation. So first step, find out what to modulate. Second step, do it in a smart way by electricity. So for example, if you have two populations of neurons and you have to only modulate one of them but not the other one, you might maybe find a way to do that using electricity, because you could, for example, find differences in the Rio base or the chronaxy of these two neurons, or just the way they fatigue over time if you stimulate in high frequency, low frequency, and so on. So that's the concept, and the conference tries to foster collaborations that lead to these concepts between researchers from optogenetics and deep brain stimulation. If you're interested in that, we're going to do another one in 2024 together, so 2 years, so stay tuned for that. But at the conference now in Geneva a few weeks back, there was one key speaker that actually made this work. So and that is Aryn Gittis, who is an associate professor at Carnegie Mellon University. And she essentially did the first step of what I just said in a Nature Neuroscience paper in 2017 with her lab. And then last year, a science paper did the second step. So in the mouse, Aryn's team found out which exact neuronal populations you need to modulate to alleviate Parkinson's disease symptoms in a potentially better way than we currently do with DBS. That was done using optogenetics. And then in the second step, she found a really smart way to replicate the exact same thing using electricity. It is quite likely that her findings will have great impact on actual clinical deep brain stimulation quite soon. So if you haven't already heard about it in the science paper, you can now hear about it here and then read her papers to get more details about the idea. So I think this is a truly remarkable and quite unique story that can also serve as a template of how to do things, especially if you have the possibility to study circuits using optogenetics, and if you also have the possibility to modulate networks using DBS. So if you've come this far in the podcast, I'm pretty convinced you're gonna love this episode, the conversation I had with Aryn Gittis. I really think this one's gonna stick out. So thanks for tuning in. Please feel free to spread the word about the podcast in general. And thank you for listening. So Aryn, thanks so much for taking part in this. At this point, I will have introduced you more formally already, so we can jump right into the science. Although, to break the ice, before we get into science, I always ask a first question about hobbies, or what you do when not involved in academia, just to get to know you a little bit. Anything you do except work in the lab?
Yeah, well, thank you so much for inviting me to participate in this great podcast that you've started. Yes, I do have a hobby that I've recently restarted. So I play the bassoon in an orchestra, and I played for about 10 or 15 years, all through graduate school. And then I kind of put it on hold when I was a postdoc, and then I had kids. And so it kind of stayed in its case for another 10 years. And just this past year, I started playing in a university orchestra again, and it was great. I just kind of rediscovered that dimension of my life. And so I plan to continue with that.
Very nice. So that's classical music, probably,
that's right?
Yeah. Super. Very cool. So and then going into the science already. So you did receive your PhD from UCSD, and then I think did a postdoc with Anatol Kreitzer at UCSF, and then moved to CMU in 2012 to found your own and start your own lab. So in your academic career, what were the key turning points that were important to get where you are now? Or maybe who were the mentors that really stuck out?
Oh, wow. That's a great question. I mean, I think I've been really fortunate to have some really fantastic mentors. I mean, my PhD advisor, Sasha Dulac, was really, I think, a strong advocate for all of the trainees in her lab. And I think, especially empowering women to, sit in the front row of talks, ask questions. She really created an environment where we felt very comfortable. And so I credit her with a lot of my sort of early formulation as a scientist. And, she also had strong writing skills and figure aesthetics that I think also were really helpful, which you don't always appreciate at the time when you're a graduate student, but in looking back, I'm like, oh, that was helpful. All right, fine. And then in Anatol's lab, I mean, he was also really a fantastic mentor that, I was always impressed. Kind of described him. I felt like he was always one step ahead of everyone in the lab. Like I say, like, oh, I kind of want to do this experiment. He's like, oh, I have those mice already, so it's all ready for you. Or like, here's that equipment that you will need to do that. It was really
amazing.
He kept it like a really exciting and dynamic place to be science. And I was also surrounded by some really excellent people. So Lex Kravitz and Alexandra Nelson, who are really outstanding scientists now running their own labs. We were all there at the same time. It was quite an adventure and a fun place to be. So that was, I had great early training as a scientist.
That's something I hear quite often, that, people come from labs where not only the boss or the mentor was great, but also it seemed to be a cradle of people that then, went on to pursue a great career, which, could be a key ingredient to success, to have also peers that are similarly driven than you are, right?
Yeah, exactly. Yeah.
Any like turning points, or even, earlier than in university, in life, anything that was important that you would think led you to where you're now?
Yeah. Maybe I'll share a story about kind of, overcoming adversity in the early years of starting my lab, because I think, this is a topic that I think is discussed more openly now, that, you have to be able to deal with disappointments and setbacks, and, science is not ever like a linear path. So maybe I'll kind of share my experience there that I think really, ended up making, I emerged a stronger person, and I think our research program is stronger because of it. So when I started the lab in 2012, I was really interested in studying the external globus pallidus, or GPE, as I'll be referring to it throughout our conversation. And and we had some transgenic mouse lines that allowed us to visualize different populations of neurons there. And so we started doing optogenetic experiments, which I can go into in more detail, but, studying how changes in neural activity shape different aspects of behavior. And, and we almost kind of stumbled upon this like really important discovery about the therapeutic potential of some of these different cells, which has really kind of shaped the direction of the lab ever since. but that initial discovery was met with a lot of skepticism by some of the big people in the field, and it was hard to get my first grant funded. It took me 9 submissions to get the first R01. And it didn't get accepted really until the paper was already accepted. I mean, it was really, we had a huge hill to climb to get our work accepted by the field, and it was really frustrating at times. I can now look back and be like, you're onto something big when people are really skeptical of it, and because if they're just like, okay, whatever, fine, then it's not that important. So the big discoveries kind of have to overcome a lot. But when I was in the thick of it, it was like really stressful. It was frustrating. Like, why don't people like, here's the data. Why don't people believe the data? And so I think kind of just talking with colleagues, both here and former colleagues, and getting a lot of just support by peer networks, also other people kind of at similar stages of their career. Like I have a kind of a network of female physiologists here in Pittsburgh that we used to meet up at friends' houses for drinks, and they really kind of helped me talk through the aims of my grant, the outline of the paper, and then just kind of give moral support. And that was, eventually the work was recognized for what it was. And so I think that even though it wasn't fun at the time, that was very formative, and it happened at a really, critical time in my career as well.
Very nice. So that it's great that you share that, because I think we definitely hear not enough about that, right? You always then see your science paper, and then it all looks so easy, but it sure was not. Since you mentioned the female colleagues, was that maybe also part of that initial problem? Did gender play a role, or would you think
I'm not sure. Sometimes when you're kind of in the middle of it, it's like, oh, it can't be me. So what are the other factors that it could be? But I think that's too easy a scapegoat. I do think that, ageism was involved. I think I was a junior guy. And like, if I had been a junior male PI, it might have been, I might have had the same problems. So I think that there is definitely, it can be hard for new people starting their labs to distinguish themselves, I think, independent of gender. And I think that's a bigger problem.
Yeah, makes sense. So I usually ask this in the end though, but since we talk about it now, any advice for young and or female colleagues entering the field?
I think like having a peer network is really important. And ideally they would be local people, because just getting together and, sometimes you just need to vent. Sometimes, it's always good to turn the venting into productivity. Like one of my friends actually bought a whiteboard for her house so that when we went over there, we could like map out ideas. And I mean, that was just just the support all around, the intellectual support and the moral support was really helpful. And, now that Zoom has become more of a thing, it's maybe a little easier to have these sessions with people not at your university, but for me, there's nothing that replaces the in-person aspect. And I think it's just really helpful to have people, even if they're slightly out, people even slightly outside your field, there are commonalities in the journey that everybody shares, and just knowing that you're not alone and being able to talk through both the good and the bad sides is really helpful.
Interesting. I just interviewed Mailon DeLong 2 episodes ago, and he said also he reported from these meetings that he had in person back in at the time where the young PIs met and discussed things. And I think it's great that you do that. I think we have not done that enough here in my environment. And so definitely should look into that more. So, but then coming to the main subject of today, we just met at the OptoDBS conference in Geneva, where you gave a splendid lecture on your recent papers. And I think the key ones are the 2017 in Nature Neuroscience and then the 2021 paper in Science. Probably there are more than that, but these to me seem like the key findings. And as we discussed, your talk and your science was one of the few examples, or maybe the only real one, that truly carried out this concept of optogenetically inspired DBS. Maybe before we dive into it, would you be able to give us an executive summary of that concept?
Sure.
What it means? Oh,
yeah. yeah, absolutely. So I do want to say before I talk about our science paper in this opto-inspired DBS, I do want to kind of give a highlight. So the previous study from
you're right. Good point. So in fact, I had it written up that way. So maybe we start with the optogenetical part. That's a good point. And the 2017 paper. Maybe can you summarize that? I totally agree. That's better to start with that. Yeah.
Oh no. Well, so I mean, in terms of just the idea to do an opto-inspired kind of DBS kind of thing, I think we drew our inspiration from a paper that was published by Megan Creed and Christian Lucier maybe 4 or 5 years earlier. So I feel like that really kind of set the stage for this emerging field, which, opto-inspired DBS. And the idea there is, let's take information that we've gained through basic research and, the study of molecules or synaptic changes or synaptic properties, and cell type specificity, like labeled lines within neural circuits. These are all things that get studied a lot in the basic research spectrum. But let's not just kind of leave it there and have it be knowledge for knowledge's sake, which is super important. But I would also like to be able to positively impact patients' lives. And so, once we had discovered, and I'll go into this in a second, but once we had discovered these therapeutic populations of neurons within the circuit, we knew that you had to go in with a very cell type specific approach, or the thing that we had discovered wouldn't work. But you can't do those kinds of cell type specific approaches in humans, or at least not. It's not something readily available. So can we, use our knowledge about the basic features of these cells, the basic biology of the nervous system, and then develop an approach that you can use electrical stimulation, which is widely used in humans, to get the cell type specificity. And so that's kind of what our idea of this, optogenetics inspired DBS is about.
Great.
So I can also go into, where does the optogenetics or opto-inspired part of this come from? Yeah. So in our 2017 paper, which really kind of set the stage for everything, we had discovered, almost serendipitously, that there are 2 different classes of neurons within the external globus pallidus, which is this, brain area right in the middle of the basal ganglia circuitry. It's been studied as being a contributor to some of the pathological oscillations or, brain firing patterns that occur in Parkinson's, but hadn't really been broadly researched as a target for therapeutic interventions. A lot of work was, most of the emphasis continues to be in the structure above the GPE, the striatum. But we were really interested in looking kind of outside of the striatum about these downstream structures that then are important for relaying commands to other parts of the circuit and outside to motor areas. So we had found within the GPE that there are these different populations of neurons that we could target in specialized transgenic lines of mice. And we also, when I was starting the lab, I kind of wanted to do some really easy project to make sure that we could use optogenetics. So the idea of optogenetics is that you can kind of trick neurons into expressing ion channels that are sensitive to light. So then you get these neurons you get these ion channels expressed in whatever population of neurons you want, and then all you have to do is shine a light in the area of tissue that's expressing these molecules, and you can turn on and off cells by using this approach. So there have been decades of research, arguing for this canonical circuit model of Parkinson's where direct pathway neurons in the striatum that express D1 receptors are kind of like underactive. Those are the ones that promote movement. And then the D2 neurons in the indirect pathway of the striatum are overactive. And it's this imbalance between the like go and stop pathways, weighted strongly towards the stop pathway that makes voluntary movements very difficult in Parkinson's patients. Yeah. And there've been, those are some of the studies that kind of inspired me to get involved in the basal ganglia research in the first place. It's like really, really fantastic work. But there was less known about then when you get outside of the striatum, like what happens. And so, an obvious test case of this, fundamental circuit model is the striatal neurons that are thought to be overactive in Parkinson's project to the GPE and they inhibit that structure. So, and there are neural recordings from, human patients and across a bunch of animal models that show that the GPE is underactive or it's, overly inhibited in Parkinson's patients. So we thought, okay, well then let's just restore activity or, renormalize activity in that part of the brain by using optogenetics. So we'll put in excitatory, channels that will drive neurons to spike more. And then we'll rescue Parkinsonian motor deficits in our mice.
I didn't even know it originated that way. That's interesting. It's a great idea, a simple but great idea. Yeah, to do that.
Yeah. And so we tried that, and it didn't work because we were targeting all of the neurons globally in that area. And at first we thought, well, that's weird. And so we did a bunch of control experiments to make sure that we actually knew how to use optogenetics, and like all of those checked out. Like we were actually boosting the activity of the neurons, and the neurons were sustaining responses during the stimulation. So there was something about the model that was wrong, and that really piqued our interest. And again, kind of drawing upon work that's been done in the striatum, where you have these different molecular populations of cells. One of them causes you to stop moving, the other causes you to start moving. If you were to globally activate all of those cells at the same time, that wouldn't be a very smart way to try to restore movement. So we thought, well, maybe we actually need to target neurons in a cell-specific way within the GPE, just like you would need to do in the striatum. And we didn't have there wasn't as much literature to draw on at the time of like, which ways should you stimulate which cells should you excite, which ones should you inhibit. And I would say that's still not, in terms of like normal movement, I would say that's still kind of an open question. But, we just started with the canonical cell type in the GPE is, enriched for a calcium binding protein called parvalbumin. Mm-hmm. It's just kind of a handy molecular access point for us to those cells. We don't really know what parvalbumin is doing in those neurons. And so we tried, okay, what happens if we just excite those parvalbumin expressing neurons? And, to our surprise and like great delight, we found that that more cell type specific manipulation actually worked. And the cool thing is that the movement persisted for many hours after doing this intervention, which was also really interesting to us because, previous kinds of interventions usually are pretty good at giving acute relief of symptoms, but then the symptoms come back very rapidly. So we thought, wow, this is maybe something really important that not only are we restoring movement, we might be changing the circuit in a persistent kind of way, inducing plasticity. We're still trying to figure out exactly what we're doing, but whether it be,
And sorry, you said the parvalbumin ones are the most canonical ones. So are there more parvalbumin than of the other type in the GPE, or is it? Do you know that?
Yeah, the number of parvalbumin neurons, just based on immunostaining, is probably about 50% in the GPE. And when I say that they're the canonical ones, I mean they're they strongly target the subthalamic nucleus, which is, sort of the textbook target of the GPE. Yeah. They also go to basal ganglia output nuclei, both the GPI and the SNR. So, kind of just in terms of where their projections go, they're like the canonical.
And do you know whether they receive more from the D1 versus D2, like input from? Can that be differentiated or? There's been a couple of results very recently looking at that, and they get input more from the D2 population of neurons. Okay, makes sense.
So that's the sort of canonical indirect pathway. Yeah.
Great, but I did not want to interrupt you. So this is really cool. So maybe to summarize, currently what you said, modulating everything in GPE didn't have an effect, but selectively PV plus that like parvalbumin plus would lead to an effect.
Yeah, exactly. So then we're like, well, that's cool, but why does it matter? Like if we stimulated that population before in our more general experiments, then why didn't it work? Like why do we have to target them very specifically? So we did some electrophysiology experiments. And one of the beautiful things of using optogenetics to excite neurons is that you don't have these electrical artifacts that you have when you do electrical stimulation. So we were able to record from the GPE, and we just stuck an optical fiber on top of our array, and then we could illuminate the tissue around where we were recording and ask, okay, when we excite these parvalbumin neurons, what's happening around them? Are they affecting other neurons near them in ways that we hadn't anticipated? And we found that sure enough, when we selectively excite the parvalbumin neurons, they inhibit other neurons around them. And that's an effect that is occluded when we just do the global stimulation. So we thought, okay, well, maybe this, inhibiting this other subset of cells is also a factor that you need to, that needs to be considered when trying to induce this persistent behavioral recovery. So we, kind of drew upon the data in our lab that parvalbumin neurons are 50% of GPE neurons, and then so who are the other 50%? And we knew that at least some of those were part of this other population of neurons that the limb homeobox 6 or LHX6 marker, it's a transcription factor. It's important for development of GPE neurons. We found that if we inhibited those neurons, then we also got the persistent rescue. But if we excited those LHX6 neurons, we didn't. So we said, aha, so there's this condition that needs to be met within the GPE to induce this rescue, where you need to excite the parvalbumin neurons. And while you're doing that, they're inhibiting the LHX6 neurons. And kind of those two things combined seem to be important for giving you this persistent rescue.
But then inhibiting LHX6 alone would also help, right? So you could, if I remember correctly, you could essentially ignore the parvalbumin ones and just inhibit the other ones, and that would also help. Correct?
That's partially correct. So it's true that inhibiting the LHX6 neurons also gives you the rescue. But if you globally inhibit everything, then that doesn't work either.
Yeah.
So you need to, so like even
selectively.
So even if you inhibit the LHX6 neurons, we found that the parvalbumin neurons don't get disinhibited. Sorry, like again, thinking about the interactions between these cells, but the PV neurons keep spiking. And so we just think that you have to kind of drive this differential in the firing rates. Like LHX6 neurons need to be inhibited relative to the PV neurons. Yeah. And there's a couple of different ways that you can get that condition to be met.
Now, I was just picturing maybe more clinically oriented listeners listening to this. So probably at this point, we should clarify how much this really matters. So what is the actual effect or like behavioral effect that you could see in these mice?
Right. Yeah. So the experiments that we're doing are all in a mouse model of Parkinson's, where we deplete dopamine by injecting the toxin 6-hydroxydopamine. This is a rather old model, and the primate equivalent is the NPTP model, where you inject something that's kind of killing off dopamine. I would also say for the clinical listeners that The way that we apply 6-hydroxydopamine, we're killing dopamine neurons, but we're also partially killing norepinephrine neurons, because we don't, you can make the technique a little more selective just for the dopamine neurons, but we don't do that because in Parkinson's disease, you get loss of both populations of neurons. Yeah. So we do these 6-hydroxydopamine injections into the medial forebrain bundle. This is the bundle of axons going from the substantia nigra compacta. Those are the dopamine neurons that selectively degenerate in Parkinson's. Yeah. And the bulk of their axons innervate the striatum. And so we do our toxin injections kind of right into the thin part of that axon bundle to kind of maximize the number of cells that we kill. And within just a couple of days of doing these injections, the normal like intense staining of dopamine axons in the striatum is completely gone. And associated with that, and we do this bilaterally on both sides. So associated with this loss of dopamine, the mice develop, very severe motor impairments, which, are similar to what patients get at like very end stages of Parkinson's. So this is like modeling a very extreme end stage of the disease. Yeah. And some of the symptoms that they have is they don't move very much. They just kind of sit there. They can move. I mean, they're not paralyzed, and yeah. In some situations, like when you go to grab them in the cage, they run away and they try to like not have you pick them up. So maybe that's some sort of paradoxical kinesia. That's something we're kind of interested in studying those circuits. But so they have a lot of trouble initiating voluntary movement. They have postural. They have a stooped, hunched posture. When they do walk, their gait is abnormal. And many of these symptoms, like the 6-hydroxydopamine model, has been used for decades, and many of the symptoms in this model are responsive to L-DOPA. So there are some limitations of the model for sure. I mean, Parkinson's is not something that appears overnight. It's more of a gradual kind of thing. But there's a lot of predictive validity of this model, specifically for the motor aspects of the disease. And
I think we can add that, STN-DBS in these mice would also have a similar effect than in humans. So positive effect towards the symptoms. So I do agree it's a really good model. And that was not really what I meant, but rather, making clear that now, in your case, if you stimulated the PV neurons and DPE probably for minutes and then stopped and while you did it, and then afterwards for hours, the symptoms were much better, like less severe, right? So I think the long-lasting thing is key, but also just the acute effect. And the same applied if you selectively inhibited the LH6 neurons in GP.
Yeah, that's right. And I will say one of the things that is striking that may also be of clinical importance to these GPE interventions is the increased movement that we see in the mice in the open field. It doesn't happen right away. So we do 10 stimulus trials. It's a little bit historical how we kind of arrived at this particular stimulation protocol, but we turn the light on to activate or inhibit one population or the other for 30 seconds, then we wait 3 minutes, and then we do it again for 30 seconds, wait 3 minutes, etc., for 10 times in a row, which is about a 35-40 minute protocol. And somewhere around the third or fourth stimulation, you start to see the mice moving around kind of even in between the light pulses. Like a lot of times we'll turn the light on for the first stimulation and the mice maybe start grooming, like they do something, but there's not like a lot of locomotion. And then kind of accumulating between trials, between light stimulus, over the course of that 30-40 minutes, we start to see increasing amounts of movement that don't even need the light to be on in order to initiate. And that's really kind of this persistent effect. And it's striking because it's a really different therapeutic kinetic than DBS, for example, which kind of works pretty rapidly.
Yeah.
And also with stimulating the direct pathway neurons in the striatum, which is also kind of a very acute thing. The mice move as soon as the stimulation is turned on, and then stop stimulating and they stop moving. So there's also, kind of this different kinetic that accumulates over time, which is really interesting. And then, we can, after we do this induction protocol, we kind of stop doing anything to the mice and just let them be in the open field. And, they continue to move around. And explore. We're working on using a more enriched environment to really say how much of these actions that they've recovered are directed versus, are they trying to run away from something, like all those kinds of things. But they become very active in the open field. But they will also do a variety of behaviors. They'll stop and they'll groom for a while. They'll sniff the ground. They'll change direction. So it's not completely normal patterns of behavior. Like you could look at the mouse and say there's something a little off with that mouse, but it's an impressive array of spontaneous behaviors that get recovered.
And how long exactly? You said multiple hours, but can you put a number to it?
So we run our experiments for 4 hours because we don't have any food or water in the cages, and so we're really only supposed to have mice in that environment for a certain amount of time. We have in some cases left them in the open field for up to 8 hours, and in those couple of mice where we did that, they were still moving around. But if we, put them back in their home cage and come back the next day and put them in the open field, they're immobile again.
Yeah.
And we can reinduce the behavior, but it doesn't seem like we need fewer stimulations like the kinetics of its onset seem to be the same as they were on day one.
Okay.
So we think that so we don't know. And we're always brainstorming. For the past 4 years, I think we've been talking about designing an experiment we can run in their home cage, so we can actually, see how much they eat and drink and sleep and just do normal mouse things after we've done this intervention. And we haven't gotten there, but that would be the idea. Yeah.
Amazing. So we probably it could be up to 8 hours, but not a day, right? We can probably say that. And that brings us, or obviously ask the question of mechanism. And I think a fair portion of your talk dealt about, thinking about the mechanism. Maybe it's fair to say that you haven't nailed it down yet, but you have theories. So I think you said something. Maybe you just give us a rundown, but you said something about the bursting activity in the substantia nigra reticulata that seems to matter.
Yeah.
Maybe, and that maybe just for the listeners, that is different in the dopaminergic depleted state, so in the Parkinsonian state, right? You have bursts there that you usually wouldn't see. Can you tell us about that?
Yeah, sure. So, I think that there's still a lot of debate in the literature about what constitutes Parkinson's pathophysiology. And, and this is super relevant for clinical studies as well. And, probably the most commonly applied clinical, biomarker of the Parkinsonian state are these beta oscillations. Unfortunately, we haven't found that those beta oscillations are very detectable in mice. So that's not a feature of neuropathology or neuropathophysiology that we can use in our studies. But there's a bunch of other things that change about the firing rates and patterns of SNR neurons. And we're particularly focused in SNR. This is probably another thing worth pointing out for clinical listeners. So in mice, the GPI equivalent is called the entopeduncular nucleus, and it's very small. I think there's still, there are people interested in addressing this, comparative anatomy sorts of questions. But I think that the GPI and SNR are sort of contiguously the basal ganglia output nuclei. And I think like parts of the GPI have gotten incorporated into the rodent SNR. So I'm going to be talking about the SNR throughout my entire study because it's an accessible nucleus in mice. Whereas the tiny GPI equivalent, which seems to mostly have retained its limbic function in mice, but not so much the motor function. Mm-hmm. So I'll be talking about SNR, but I would assume that many of the things I'm talking about will sort of apply to GPI in humans and primate. So in terms of, what are the features of neural activity that change in basal ganglia output nuclei? Certainly there are changes in firing rates, but we found that those are variable across studies. And what seems to be much more reliable are these like dramatic changes in firing patterns. Neurons after dopamine loss become really irregular. And one feature of that irregularity that we found was particularly affected in our study is this kind of burst firing, where the neurons they just sort of go. We used something called the Poisson surprise test, where like when spikes are more clustered in time than you would expect, then that's kind of classified as a burst. So this was the feature of neural activity that we found was particularly affected by our therapeutic interventions. So in the Parkinsonian state, you've got a lot of these, pathological bursts going on in the SNR. The theoretical idea that has been postulated by others in the field is that when you have these irregular patterns of neural activity, that just kind of sends like a noisy signal that confuses other downstream neurons so that they can't appropriately process motor commands. So the basal ganglia is now spitting out this noisy signal that's screwing everything up. And that's why for a long time, original therapeutic strategies for Parkinson's was to just cut out the output nucleus of the basal ganglia. It's like, you're better off without it, without this noisy signal it's spitting out. So we think that these bursts are at least, one reliably quantifiable metric to sort of get a handle on this, pathological noise that the basal ganglia is injecting elsewhere.
And you record them electrically, right?
That's right. Yeah.
Okay. Mm-hmm.
That's right. And so we found that if you just look, there's some neuron, and we're still trying to understand who these neurons are, but there's some neurons in the SNR that get particularly bursty after dopamine depletion. So we just kind of said, if you just look at the neurons that are kind of like 2 standard deviations beyond the mean in terms of like how many bursts they have. And those were the ones where during our therapeutic stimulation, you could actually see the bursts in those neurons disappearing. And then after, inducing this therapeutic, going through the whole protocol, if you follow the activity of these cells for up to 2 hours. It's a little harder to follow these cells for very long periods of time.
Sure.
But for at least 2 hours, I think maybe in some cases we look for 3, you don't see these bursts returning, at least not to the same levels as they were in the before interventions. Yeah. So our hypothesis is that somehow these interventions that we're doing are preventing, they're minimizing these aberrant firing patterns that have emerged in the depleted state, and they're helping to restore those patterns at least a bit more towards the healthy condition. Yeah. And so maybe the basal ganglia is still sick and not working quite right, but it's at least not being as destructive to motor propagation as it was in the full depleted state. So we think that might be one of the things that we're doing. And interestingly, when we looked at what happens to these bursts in the SNR during global GPE stimulation, which was something that didn't work therapeutically, these bursts are still present. So there's at least like a correlation between the severity of these bursts and the therapeutic efficacy of our technique.
Interesting. But you would say could they be the same thing as beta bursts in humans, just because frequency or probably hard to say, but I mean, both are bursts. That's why I'm asking.
Yeah. So I don't know.
We don't know. Okay.
I would love that to be the case. I will say though that there was another study. I had a computational student in my lab that was a joint student between me and John Rubin. So John Rubin is a mathematician who's been really interested in just understanding neural dynamics that can lead to pathological oscillations in bursts.
Yeah.
And what we found is that probably a lot of these bursts, at least in the super bursty neurons, are related to, they're in kind of a delta frequency, like a 1 to 4 hertz thing. And actually, if you go back to the clinical literature, some patients have delta oscillations, but no beta oscillations. And there's like fewer patients have beta, but no delta. So it seems like these delta oscillations may actually be really important, and they've kind of somewhere along the line, they just kind of got forgotten about. I think they're a little harder to record than beta just for some technical reasons, but those might actually be a biomarker that translates from mice to humans.
Interesting. Might make sense to look into or you probably know the literature from Andy Sherrod in London. I think he's recording beta in rodents quite a bit, but it's probably rats.
In rats, yeah. It's weird. I wouldn't have thought there would be many differences between mice and rats, but I've actually talked to some colleagues that have studied beta oscillations in rats. Yeah. And then when they tried to do some of the same studies in mice, they're like, mice just don't have very strong beta oscillations.
Interesting.
Yeah.
Didn't know that. Okay. So great. So we had in Geneva at the conference, we had speakers dinner on a boat. Was really nice. And then Julian Neumann, who is also a co-host here and was interviewed in episode 16. He asked, I think, an important question that could be confusing in your line, or like for readers. I think he asked whether either of these two populations were archipallidal or whether both were prototypical. Could you maybe define even what that is, and then answer the question?
Yeah, sure. So there's another subtype of neurons in the GPE that I don't think that we've been studying very much, are these archipallidal neurons that were first described by Nico Mallet. Maybe when was that paper? Like 2008 or something. So it's been a little while, and these are kind of non-canonical cells where instead of going to the STN and like downstream basal ganglia nuclei, they project back to the striatum. I do want to say, because I worry that there's one detail that is getting glossed over. So actually, many GPE neurons, including prototypical neurons, go back to the striatum. Gotcha. But prototypical GPE neurons that project to the STN, for example, their projections are very specific for interneurons in the striatum. Those interneurons are like 5% of all striatal neurons, and so those projections don't appear very densely. Okay. But most GPE neurons do make some sort of projection to the striatum. Okay. But these archipallidal neurons are unique in that they don't go downstream; they only go upstream, and they form really dense net-like projections, which is where the name archipallidal comes from. And they target kind of medium spiny, the main class of striatal projection cells directly.
They inhibit them as well.
They probably inhibit them. Yeah. The physiology is not as clear. The anatomy is super clear. Yeah. The physiology does not match the anatomy, which I find really interesting. But yes, I think the idea is that they can broadly inhibit striatum. Yeah. And these archipallidal neurons that you bring up, they seem to be really functionally distinct from other GPE neurons. And these archipallidal neurons get input from the D1 striatal neurons. I think this is a question that you had asked earlier in terms of like who the parvalbumin, like who in the GPE, who's getting input from where. Yeah. And so the archipallidal neurons get input from the D1 cells. But we also found that some of the LHX6 neurons that we need to inhibit, in fact, we think that the way we're able to inhibit LHX6 neurons when we do electrical stimulation is that they're also getting input from these D1 cells. Okay. But the LHX6 neurons are not archipallidal in that they don't broadly inhibit, send really dense axonal projections throughout the whole striatum. So so I think that there's still a lot of kind of pathway complexity in the GPE that we don't understand yet. I will say we haven't completely ruled out that the archipallidyl population, maybe it's playing some role in the persistent rescue that we're inducing. We've tried a couple of times to look at that, and I would say our results are ambiguous, and it's certainly never been as robust an effect as when we target the LHX6 and PV. So
but maybe to state clearly, as you said it on that boat, that both of your neuron types are prototypical, so none, like they are not archipallidyl neurons, because I think many people that, like me, that don't know too much about the GPE would think, oh, two classes, that must be, prototypical versus archetypical, which is not the case. I think that's the key point, just to clarify. That was helpful for me to understand this better. So I think it's a great point to briefly summarize. Very simple terms. What you found is with optogenetics, exciting PV parvalbumin neurons is a good thing. LHX6 should be, inhibited. Mm-hmm. So you kind of want to tip the balance in activation towards parvalbumin in the GPE, and that leads to these long-lasting effects that take a while to build up, that could be up to hours, and that's really clinically relevant because it stops Parkinsonian symptoms. We're brushing over many things, but just to get a summary at this point. And I guess now we can go into how you put your electrophysiology hat on, as you said in the talk. So and maybe to reiterate, in episode 20 of this podcast, I interviewed Christian Lüscher, who is the main organizer of the OptoDBS conference, and who, together with Megan Creed, as you said, who was also in Geneva, published their 2015 science paper entitled "Refining Deep Brain Simulation to Emulate Optogenetic Treatment of Synaptic Pathology." And I think you had mentioned that this concept was important. I'm sure there's your own contribution. So I just want to say this is such a great example of a concept where, as far as I understand it, you try to understand something as precisely as you just outlined with optogenetics in the mouse. And then the next step would be, now that you know what you have to do, is how can you maybe do it with electricity in the mouse first? Because if that works, you can then also go into humans, because there we don't have optogenetics yet. So let's look into your 2021 science paper. You said you put the electrophysiology hat on, but why and how? What did you do?
So we had this result from optogenetics in mice, and I'm like, wow, this is really cool, and it would be great if we could implement this somehow in humans, so that we could extend, even if you still need to have DBS, some sort of intervention every day, if instead of leaving the stimulator on, if you could just, give, 30-minute treatments twice a day or something, and then keep the circuits running, that could really be transformative for how we deliver DBS. So we wanted to know, is there some way, and we know from our stuff, if you globally affect all of the neural activity together, you're not going to get these persistent effects. So can we somehow deliver electrical stimulation in some pattern or in combination with pharmacology? I definitely thought we were going to have to use pharmacology to give us the same pattern of cell activity that we got with optogenetics. And so we started doing, bread and butter slice electrophysiology experiments, and just asking, when we're doing electrical stimulation, how do the neurons respond? By doing this in acute brain slices, we're able to get really good signal to noise. So we actually were able to kind of get rid of the stimulus artifacts and look at the firing of the neurons during stimulation. And then we could actually go one step further and ask, not just, well, how are the neurons spiking, but what are the actual synaptic currents that these neurons are experiencing that's driving their firing? And when we looked at those, it was really striking that the LHX6 population was getting this, this huge inhibitory input that wasn't present in the PV cells, but it decays really rapidly. So as soon as you've been stimulating at 100 hertz for over a second, this inhibitory current on the LHX6 neurons is gone. And then there's just this, weak, low-lying, probably slight excitatory drive. So we thought, well, and this huge inhibitory current wasn't present in the PV cells, or at least to a much less, a lesser degree. So we said, well, right here, like even without using pharmacology, which would be great if we don't have to do pharmacology, because that lowers the bar for what you can try and translationally.
Yeah.
Maybe all we need to do is keep our stimulations brief so that we don't kind of override this intrinsic asymmetry that's naturally present. And so if we just kind of deliver stimulation in line with what the synapses can keep up with and handle, then maybe that's going to give us the cell type specificity. So we can basically take advantage of this natural architecture of the circuit that we just hadn't appreciated was there before because we hadn't really looked. And so when we went ahead and just tried stimulating in short bursts. So, I think the original thing we tried, at least in our slice experiments, was 1 second. And then we would just kind of do this every, 20 seconds or so. We found that the LHX6 neurons were reliably inhibited and the parvalbumin neurons were reliably excited during these brief bursts of stimulation. And so that was kind of the start of how we discovered this opto-inspired DBS pattern.
Really cool. And you said that if you would stimulate longer than a second, the synapses would fatigue in the LHX6 neurons.
Exactly. Yes.
So that, and then the idea is to burst them. That's really cool. And it's 100 hertz with pulses of, is it also 60 microseconds or something?
Yeah, it was 60 microseconds, and in our slice experiments, I think it was around a milliamp of amplitude that we were using. Yeah, cool. That's right.
And then in vivo, you also tried that.
Yeah, exactly. So we tried. Did a couple of other experiments, and actually I will say one thing before we jumped in vivo. At the time when we first saw that the LHX6 neurons were getting this inhibitory input, we're like, well, that's awesome, but we didn't know where it was coming from. Mm-hmm. And that kind of mattered, because when you start to think about, well, how do you go from a brain, almost like a two-dimensional brain slice to actually stimulating in an intact brain in vivo, where did we want to put our stimulating electrode? And so this is when we did these experiments to find that that it seems to be the D1 striatal neurons that are selectively targeting the LHX6 cells, not the PV cells. And that was where the inhibition was coming from. So even though we're not stimulating in the striatum, and in slice we were stimulating kind of in between the GPE and the STN. We were kind of antidromically activating these axons of D1 cells that were going through that area on their way to the basal ganglia output neurons. So in between STN and GPE, or that's where we were in. That's where we were in slice. Okay. Yeah, we were. So it wasn't at all clear that, like, if the inhibition was coming from striatum, which the striatum is known to inhibit the GPE. Sure. It was. But from the D2 cells mostly, and their axons like stop at the GPE and don't continue to go downstream. So it was we were like, well, how would we even be activating striatal inputs because like our stimulating electrode isn't in the right place, and then we kind of rediscovered. I mean, these collaterals from direct pathway neurons that stop in the GPE on their way through to basal ganglia output neurons have been really well described anatomically, and even before the establishment of this canonical direct indirect pathway model. But they were just sort of confusing, and so and like a pain to think about. It's like let's not even go there. but they're definitely there, and we think are very important for being able to recruit the cell type specificity. So anyway, and if you look at where their axons go, at least in mice, this suggested to us that we might have better luck getting the cell type specific response in vivo if we put our stimulating electrode closer to the the GPI equivalent rather than putting it in the STN. And so that's what we went ahead and tried in vivo. And this is like an example of where there's like an obvious experiment to try that will like make or break your entire hypothesis. And I was so nervous to try these experiments. I was like, oh my God, this is never gonna work. I don't know. It's just like, we'll see what happens. And it was, it was just incredible. Like when we took the same mice, they were dopamine depleted. And on one day we would give them conventional DBS, which is like continuous 100 Hertz for 30 minutes. And then on the next day, and we would, kind of reverse like some mice got conventional one day and the next day got burst and vice versa. And then on the days where the mice got burst DBS, during the period of stimulation, the movements looked about the same. But after we stopped stimulating, these mice that had gotten burst DBS kept moving in the open field, just like what we had seen with optogenetics. That was a really exciting I would say
I can only imagine it. I was about to ask for her eureka moments in the end of the podcast. So that surely was one, right? So you have a prediction, you've seen it in the slides, it should make sense. But then behavior is a whole different thing. A living mouse is a whole different thing. And what you said, I'm sure it's not going to work. I can totally feel that. You always doubt yourself, or usually people doubt themselves, right? And then, but then it did work. That's amazing. And one detail you said, sorry, maybe before we go into that, did you celebrate like as the lab, or for beers? What did you do? Did you dance?
It was the realization that it was working was it was too slow to be the perfect eureka moment, because you see it in one animal and it's like, is that reproducible? I don't know.
Sure. Yeah.
So it was kind of a gradual thing that when it became clear that this was going on, it was kind of like, this is really big. So
One thing that might seem to like a detail to you, but not to me, is that you mentioned that the electrodes were actually in the GPI equivalent, so in the entopeduncular one nucleus. So I didn't know that. I think many people don't know that, maybe including Julian, that we always talk about, we have to kind of get electrodes into the GPE, but it sounds like we don't.
Yeah, I think, in humans. This is something that this is an important translational question that kind of needs to get figured out. And so we're collaborating with Rob Turner, who does monkey research with DBS, and it like really trying to figure out where is the sweet spot in primates. There's this kind of border zone between the GPE and GPI that I think Jerry Vitek's group has kind of identified that, like, even not with burst stimulation, just generally, that's like a really good area to stimulate.
I think many people have. Phil Starr has a paper on it. Julian has a paper on it. So this border in between, maybe Jerry was first, so it can definitely be. But I think you're totally right. So these I sometimes even think maybe the border cells play a role, but. Yeah. Any thought about border cells? Did you look into that at all?
No, I have not looked into them.
Okay. Yeah. But great. So that is so exciting. So again, maybe to recap here, you did use a genius way of, you realized you get an inhibitory effect in the LHX6 neurons. That was one of the possibilities you had, right? Either upregulate the PV or downregulate the other ones. And you got that, but it depletes after 1 second of 100 Hertz stim. So then you just say, hey, let's just always do 1 second bursts, right? And then it still is there. And that worked in slice and then worked in mice as well. So I guess that is the nearly complete story of really using optogenetics to inspire DBS. I think that the final piece is really to do it in humans as well, right?
For sure, yeah.
So, and I think you, maybe before we go into that translational aspects, I think from your talk, I remember that you were able to map the findings to D1 direct pathway neurons, and you already said something about that. But you also used inhibitory dreads to confirm that further. Is that, and could you maybe to begin also to define what a dread is?
Yeah.
How you use it?
So I can never remember it's an acronym, and I can never remember the full, like designer receptors, so blah, blah, blah. But, and I don't mean blah, blah, blah to be glib. It's like an amazing technology. I just can't ever remember all of the words. But the idea, I mean, similar to optogenetics, the idea is to put a molecule in the cells that will allow you to control their activity. And so a dread is it's a receptor.
You design the receptor and the drug, right? So I guess that's the idea.
That's what, you need, so instead of being an ion channel like optogenetics, it's a receptor that's usually G coupled. So G proteins can, kind of set the tone of a neuron and make neurons generally more excitable, less excitable. But the ligand that activates the receptor is a drug that's not found in mammals, and you just kind of inject it, and you can either inject it IP, like systemically, or you can infuse it into the little part of the brain that you're studying, and that will activate these receptors that you've put into the cells of interest and allow you to kind of blanket their activity or kind of boost their activity accordingly. So the experiment that we ran to, we had a lot of correlative evidence that the direct pathway neurons were supplying the inhibition to LHX6. To really test that, what we did is in slice, we did our electrical stimulation and showed that we were inhibiting LHX6 neurons, exciting PV cells. And then we, in the same slice, we washed on the ligand that activated these inhibitory dreads that were expressed in the D1 cells. So basically, we washed in we changed the condition so that now when we electrically stimulated, the D1 cells weren't activated, and that got rid of the inhibitory response in the LHX6 neurons. So that was kind of the direct causal experiment.
Really cool. So so many ingredients. This is so impressive, and that gets you then, I guess, a science paper, including it. The methods alone don't make it. You also have to find something cool, I guess. So as I mentioned, the last step would be to confirm these findings in humans. And since it's a science paper, I'm sure many people knocked on your door. Is there somebody that's going to do this in humans? And you already mentioned the Pittsburgh group doing it in primates and looking into it further, but are there? So there's a group in Pittsburgh. Nestor Thomas and Don Whiting are some of the key neurosurgeons there. I think that's ongoing. I think that they're still kind of trying to identify patients that are willing to give this a try. We also have recently been collaborating with Nader Portian at UT Southwestern, who has tried this in a limited scale in patients as they're still in the operating room. And so not a condition where we can really study is it inducing persistent effects, but just what are the effects on the nervous system? And it does seem to be promising, at least in terms of this is brand new data, so it's not super far along yet. But I think it's just sort of honestly one of the main things that the Pittsburgh group is going to start with is just a tolerability study. Because I think we're not even sure if you have a stimulator that's going on and off, is that going to feel really weird to the patients? Sure. Is that going to induce some weird motor effect? So that's kind of the step one. And it seemed like the burst stimulation was well tolerated in the patients, at least in this, super early days of the study. Very cool. So if let's say we flashback to the future, 5 years from now, everybody does DBS that way. How would you feel?
Yeah, I mean, I think it would be great.
Sure.
You know, I would say that the pie in the sky for where we'd love for this to go is, again, we still don't know why the effects are long lasting. Yeah. You know, one of the things we want to look at is it inducing some sort of arousal effect that is allowing patients to stay in a state where they can move. And, you know, that could be some sort of neuropeptide or some factor that gets released. You know, another possibility is like, are we actually driving therapeutic plasticity that is helping to keep the basal ganglia out of this pathological state? And I would say that that's really would be like the most exciting answer to what we're doing. Because I think really, you know, to really marry where we're at with basic research and clinical application, we want to understand not how to do, you know, palliative care to slap a Band-Aid on the symptoms. We want to fix the circuits. Yeah. And so that's really, you know, where I hope that this is going. And we've got experiments going on in the lab right now to test that hypothesis.
Amazing. Yeah, no, I totally agree. And I guess you mentioned before, if you could, you know, have the advantage of switching the stimulator on twice a day only, that would be cool. I agree, you'd use less battery, but it might be, you know, in the end for the patient, just the same thing as continuous stim. So I totally agree. It would be much cooler if it were a different effect that might either, you know, suppress symptoms better. That could be true, right? Without less fluctuations or something like that, especially in the end stage of Parkinson's, or of course, disease modifying to, you know, slow progression of the disease. So either possibilities, I think, could be in on the table. That will be really exciting to see. So really cool. Maybe I'll ask one last general question before we end up with some rapid fire, you know, quick questions to conclude. I've taken a lot of your time already, but it's a silly question maybe, but maybe you have some thoughts. What is the role of the GPE to begin with? You know, is it even possible to say something like that? Is there a role for the nucleus?
I would say there's definitely a role for the nucleus. I think that we are still, as a field, trying to figure out and nail down what that is. Yeah. And I also think that the function of the GPE changes in the depleted state. So in a disease state, it's doing something different than what it is in a healthy state. So we're that's one area of research in the lab is to try to get at that. Yeah.
Okay. That's what I'm rather alluding to in the healthy state. You know, what did the brain why did it come up with the GPE? So there are probably many reasons, but and it's probably hard to say it in simple terms. And you're right, we don't know it. I guess one model where it sticks out that I really like is Hakai Bergman's, who was also on episode 17 here, 3-layer model of the basic ganglia, where, you know, he essentially put striatum and SDN on the top layer, and then the output nuclei, and then in the middle is just the GPE. So it's just the middle layer of the basic ganglia. That's not a function, I know, but in terms of, you know, just artificial neural networks, I sometimes wonder that there you could really picture it as a 3-layered and you know, neural network that does some sort of processing. And obviously, I'm sure there are more layers than that, although, you know, these nuclei are quite homogeneous. But do you ever think of the basal ganglia as a neural network in terms of the AI neural network at all? Is that, do you see it like that, or?
So I would say, so my background is not in computer science. Yeah. So I would say never in a meaningful way have I really thought about it like that. But, you know, I'm inspired by the work of others that think about it that way. Certainly being at Carnegie Mellon, it's a school that's known for computer science and engineering. You know, it's at least on a high order kind of theoretical scale. I definitely do think about that. Yeah. And, you know, and guys, redrawing of the basal ganglia circuit, which I still need to just start using in my talks. You know, like the GPE is up at the top, and it's like the STN and striatum are kind of feeding in. Yeah. I think in a more concrete way, there's a study that we put on bioRxiv in the lab just recently, and we're kind of working through the revision process, that suggests that there are these different streams. There's, striatum to GPE and then downstream, but also this, STN input, thinking of the STN as like another input structure of the basal ganglia rather than, like a downstream thing of the GPE. Yeah. And striatum and STN are probably conveying very different types of signals to the GPE, which is then processing, trying to make sense of that, and then deciding what goes on downstream. So I think that there's a lot to discover about the GPE. Yeah, it's more than just a sign inverter, like it's been predicted.
He said in the podcast that the the STN, if you compare it to a piano, the STN might be the pedal and the striatum is the melody. So I really like that, because to some degree, if you really think of the GPE being the converging substrate of that, the pedal could rather do something more qualitative, stop or not. We know the STN kind of acts like a brake in many occasions in the brain, I think, but the real information content could be more mediated by the striatum, which is obviously much bigger and has much more complexity and so on. So anyways, we have to find out. So for me, the GPE is just a mystery structure. It would be, if somebody had asked me, I would have said no clue. I know it does, wire to there and, receive input from there, but something like and there's never a single role about a brain structure. It's always much more complex, but maybe this break for the STN is a good example where maybe you could attach a role to a structure that at least captures some of the rich the rich processes that it has, and such a similar role for GPE. We probably both don't know. So interesting. Wrapping up maybe with some rapid fire questions. So we talked about eureka moments. If there is another one, please share. If not, was there ever the downside, like the opposite, something where you said this was a complete waste of my time, or where a failed experiment or something where things did not go as planned?
We've had plenty of failed experiments, but they've usually been for technical reasons. I would say one of the technological boom that's happening right now in neuroscience is both exciting. It's like equal parts exciting and frustrating, because there's a bunch of tools that get published that work in a very particular system, but then don't generalize to other parts of the brain. So I would say, we've definitely invested quite a lot of money in getting the tools, reagents, setting up the experiments, and then it's like, oh, the tool doesn't express here, or whatever it is. So certainly there have been many frustrations like that. I mean, knock on like every piece of wood around me, like like I haven't had too many hypotheses get like completely shot down recently, like once we've been testing them. But I will say also, if there's any like young scientists listening, when I was a graduate student, like nothing that I tried worked. like the techniques worked, and I did a lot of really, foundational work as a grad student studying the pacemaking currents that allow that, kind of endow different firing properties of vestibular nucleus neurons, able to spike fast or slow, and thinking about why that might be important. But none of the really big, exciting stuff that I tried as a grad student worked out. And all of my graduate work was published in the Journal of Neurophysiology. I think I got rejected from J Neurosci like 4 times. And so certainly there have been many times in my scientific career where I've like tried something and it didn't work, but yeah.
So can you derive good advice for young people entering the field from that? Or you keep persistent, but other things that you...
I think that the best piece of it, there's all kinds of different ways to measure success in science. And I think the most important way to be successful is to design experiments that will teach you something. And then, what journal it gets published in is other people's perception of how important that discovery is. Yeah. But if you're doing rigorous work, and not everyone does, but there's, I've always strived to like whatever I put out, maybe someone will figure out there's a different interpretation, but people will always be able to look at the data and be like, yeah, that was a really clear test of that particular hypothesis.
Yeah.
And then, papers that get published in, journals that are smaller, sometimes when they're really rigorous, those are actually the more important papers that are going to keep moving the field along. And it takes decades of these, seemingly incremental advances to actually make the big, discovery that generates a lot of enthusiasm. But there's many small things along the way. And if you only ever try to hit home runs, that's not really like a good way to do science.
Great. Sounds good. So the future of the field, neuromodulation, what do you think is coming? What are next things, or also in the distant future? What would we talk about in 10 years?
Yeah, so I'm very excited about the potential of neuromodulation moving forward. I think, especially as we start to link, how is there's, many decades of experiments in slice electrophysiology studying how electrical patterns drive plasticity in neural circuits. I think that's essentially neuromodulation's potential. Is you can go in and apply electrical stimulation, and somehow we just were never able to kind of link those things. Like in the clinic, we've been really concerned about where to put the stimulating electrode. But I think now as sensing technology is getting better, you can sense, you can record neural activity or, derive these other markers of neural function. While you're stimulating, we can start to become more sophisticated about the patterns of stimulation, and that can be used to try to resculpt neural activity and really restore the activity of circuits in disease. So I'm very excited about that. I think also, advances either in TMS or other like ultrasound, like non-invasive interventions. I don't know how far we are with those technologies for deep structures, but I think that, if patients don't have to have this choice, like undergo this really invasive surgery, and it will probably really help, but we can't be totally sure. Like, I think the lower the bar for the patients to have to make this like life-changing decision is going to allow us to really start to push the boundaries of like just how much can we do with neuromodulation.
That's a great idea. Absolutely. So any experiment or opportunity you think that's missed? So something you don't want to do yourself, but somebody should be doing? I like the translating what we've done in mice into humans, I think is going to take quite a lot of work, and it needs dedicated collaborators on the human side. And I think that there's a lot to discover. So I don't think it's like one project. I think there's like many goals to do there. So I think that there's. And as a field in total, is there something we should be doing more, but we aren't? So missing opportunities currently. Not to like sell the OptoDBS meeting, but I'll do it a little bit. I think that it's a fantastic meeting because it really tries to bring together basic researchers and clinicians in meaningful ways that I don't think a lot of meetings do. There's basic science meetings and there's clinical meetings, and there's a couple examples where they're integrated, but not nearly as many examples as I think would be helpful. And it's hard. The clinical language is very different from basic science language and vice versa. So it's a really hard bridge to build, but I think meetings like OptoDBS are really more of that needs to happen, and I think that will really help. Basic researchers need to know what's possible in the clinic and what are the real life problems that clinicians are facing, and clinicians need to know, need to stay on top of the science and we found the cell. Do you know how to target that cell? And, good point. And I'm really glad you mentioned that. So I'm sure Christian Lucia will also be really happy if you're saying that. So So I totally agree with you. At the meeting in Geneva, I still sometimes thought, we could still do better in terms of exactly the spark transferring between opto and DBS fields with, people like me having a hard time sometimes to follow the talks of the optogenetics world where, you talk about, especially the methods talks on optogenetics were quite out of my reach, with differences in the, optrodes or in the, the molecules and so on, which are obviously normal for you guys. But then the other way around, it might have seemed similar to, let's say, a postdoc that only did optogenetics in their work and then not even knowing what Parkinson really is, or so, and maybe what the limitations of DBS are or what DBS does. So we're going to do it again together in 2 years. So I'm already thinking vividly about how can we make it a bit better, and maybe you have some thoughts. So we shouldn't just say, hey, please everybody dumb down your talks, because that's also not so interesting then, but maybe target them a little bit more for a broader audience or for a clinical audience from the optogenetics side. Or do you have thoughts on what could improve such meetings further? You were one of the key examples where it worked, right? But
it's a hard challenge. I mean, I could see something like, if there was some sort of panel discussion, or maybe even like a small group discussion where you have some basic researchers and clinicians, and they have to come up with 3 or 4 questions, here's the problem in the clinic that clinicians wish basic scientists could solve. And the basic scientists could say, here's what I wish you guys could do in the clinic. And then at least, even if there's no solutions, at least kind of present those groups, very small focused conversations. From the meeting could emerge, the grand challenges, here's the top 5 things that emerged as the grand challenge for bridging the gap or something like that.
That's great. So I also thought about panel discussions, or at least one or so, or maybe each day a small one or something to actually get people to talk about this a bit more informally after the talks, or, inspired by the talks. So that I agree would be great to do. Yeah. Let's see. So what I can also say is I attended only twice so far myself, and the first time was worse, right? So even by attending and not understanding too much the first time, 2 years later or 3 years later, I came back and I did understand more, right? So you get that, you have that. I mean, I also, of course, got a little older and, more into the literature, but also it might have, planted a spark in me to read more, think more about optogenetics in the first place, which that alone could be an effect, right? That is a more long-lasting, but not immediate effect, but that could help just to, if not, I could, go through my whole academic career without thinking about the mouse at all, right? As a human researcher, and not knowing the anatomy of the mouse brain and so on. But going to these meetings helps you, push yourself a bit to get into it. And somebody like you who speaks both languages is really unique, and that's really cool. Anyways, last question, maybe anything we did not discuss that you wanted to discuss, and anything you wanted to mention or so. We've covered a lot, but is there anything else you wanted to talk about?
No, I think we've hit it all. I mean, I'm glad that at the beginning we got to talk a little bit about the just the path to, having a success in science often means that there were many obstacles that you had to overcome. And I really think that that's an important message to make sure that everybody hears. But also, I would also just say, again, if there's any young scientists listening that, I've almost heard too much pushback, especially on Twitter. It's like, oh, academia is so hard, and why even try? And that it really is fun. I mean, it's moments where you have those eureka moments, or that you're working on a really great project that maybe is only really interesting to you and 8 other people in the world. But it just, the thrill of it is like unparalleled. I think I really love being a scientist and being able to have the freedom, specifically in academia, having the freedom to like go after these questions that you think are important. It makes everything worth it, and I think it's a great a great profession.
That's a really nice thing to say. Absolutely. There's in fact, sorry about that. One more question I noted down that I forgot to ask, but really wanted to ask you, and that came up during something you said. You said that you in the beginning you thought in your experiment. So it's the last question, I promise. You thought you would have to add some pharmacological agent to the stim to make it work. In a way, for example, you could block the LH6 ones and then only stimulate everything, and that would lead to a parvalbumin net increase. So is that, and that's something I just personally recently only heard about as a concept. So do you think that is a big future field where we can, if we understand things better and then do DBS, but we have to add a pharma ingredient, could that become a big thing in the future, do you think?
Yeah, I think absolutely. I think that we don't know what pharmacological agent to add. But I think for sure, like combining, modulators to target certain molecular pathways, again, as drugs and knowledge of basic science get more involved, maybe an electrical stimulation by itself won't drive plasticity, but if it's coupled with when you've put neurons in this heightened plastic state because of some drug you've added, then you can get the plasticity. I think, these are definitely directions that I think we'll be talking about more often over the next few years. Yeah.
Super. Bonus question. Sorry about that. And thanks so much once more for your time. This was exciting, very cool, a prototypical story that you have there that could not only change Parkinson's disease, but also shows how great it can be to combine all these methods and come up with a coherent story. So thank you so much for taking your time once more to talk to us.
Yeah, thank you for doing this. This was really fun.